Bonk. All right. So hello, welcome everyone. Not all of whom I know. The main thing we were trying to do today is that Herbert would like us to curate, excuse me, a bunch of SED-ML models for biosimulators and biosimulations, so that we can say to the NIH, hey, we've curated all these things that can be done. There are tools to do it. It's straightforward or whatever. So in order to do that, like if the more people that are doing this, the easier and faster it'll be. So this is sort of going to be an overview of how we can do it and also why. And since there are a bunch of people and I don't know how everyone's level, I thought I'd start with just an introduction to everything, because while everyone's familiar with at least one part of it, I don't know if everyone is familiar with all the parts. So,
Lucien, before you go too far, were there a list? Is there some description of the setups we need to do? You know, what needs to be installed?
I don't have. I mean, it's in general all you should need is Python and some GitHub repositories or something, or not even GitHub repositories. So in general, I'm going to start. I'll do the sort of introduction to everything. Bilal will tell people about biosimulations, the website, and how to work with it, and then I'll go through a Python script that I wrote that sort of is illustrative, I think, of how you would grab a model, do stuff with it, and
okay, excellent. Thank you.
Let me share my screen. I think, oh yeah, I should start this. Okay. There we go. All right. So curation overview, all the different parts. There are all sorts of bits and pieces of the overall sort of ecology of the system. There's SED-ML and SEL-ML, there's SED-ML, Combine Archives, Biosimulations website, Delirium and Composite, and Biomodels itself. So this is going to be overviewing everything. I'm going to focus a lot on SED-ML just because that's the main thing. As SED-ML curators, you're going to be as sort of the experiment curators, but I'll fill in everything else and also just sort of give you a rough sense of why we're doing this in the first place. So SED-ML and SEL-ML are model definition languages. They were both started around the year 2000 in order to encode models. The difference between the two is that SED-ML has semantics sort of baked into its structure with reactions and species and compartments. And SEL-ML is deliberately just the math. All the semantics are in annotations in their models, and all the core elements are MathML associated in different ways. SED-ML has a sort of a larger ecosystem of simulators and editors and viewers and things like that. SEL-ML has fewer tools, but it covers a wider range of modeling areas. You can model a beating heart in SEL-ML, where you can't really in SED-ML. So it's generally, SED-ML, which we're going to focus more on, is the simulation experiment description markup language. So while SED-ML and SEL-ML tell you what the model is, SED-ML describes what you do with it, how you manipulate things, what your simulation is, what your analyses are, everything like that. SED-ML was produced. Its sort of main driving cause, the thing it wanted to do the most, was to be able to reproduce figures in papers, and that's exactly what we're going to be doing. Is literally taking a model, represents a model from a paper. Paper has a figure in it. We want to also get SED-ML and the model to produce that figure. Other things that SED-ML is useful for is to share a model experiment with other researchers. You may want to share a model experiment with yourself in 6 months. If you set things up and store them as SED-ML, you can then come back to them and sort of more easily understand things than just a computer program, perhaps. You can also share a model experiment with someone without direct access to the tools you used. So maybe someone's on a different OS, maybe someone wants to run a different simulator, a variety of other things. Any computer in over 10 years is going to be different. So like people in the far future in quotes, in computer time, you should be able to like produce the same thing that you did computationally on that. So another way to solve that problem is to use workflow languages and Docker images. And we're using SED-ML instead. The reason we don't is, I mean, sometimes workflows and Docker is the right solution. But workflows and Docker images really focus on repeatability. You want to do exactly the same thing in exactly the same way. I should probably explain what a workflow language and Docker images are. A workflow language is essentially a script that says, run this command, run this command, run this command, run this command with these command line arguments. It's very prescriptive and precise in exactly what it does. And Docker is a repository of virtual machines where you can download a particular thing like a Ubuntu 2020 with these particular things loaded onto it, run a particular set of things on it and stuff. So it's great for repeatability. For reproducibility, what you want is not necessarily to have to use exactly the same setup that someone else did, but instead use something else. Again, different OS or a different simulator. There may be aspects of a particular simulator that gave you sort of quirky results or gave you results that were dependent on that particular simulator because of something it does that may not be precise and exactly, it may not relate to the scientific problem that you're actually trying to solve, but is related to something else. I don't know, precision in their numbers or something. So when you reproduce something, you have a better belief, you have a bit more evidence that your conclusions are true in general. SED-ML is all about the semantics of the experiment that you're running, which gives you more flexibility and more extensibility. If you describe the semantics of what you're doing, you can then say, oh, okay, I want to do it slightly differently. I want to do a new analysis. I want to do something. I want to take the figure that I found in the paper, find out what happens if I run it for another 2 minutes or something. So you're hopefully giving more tools to the scientists that are using the SED-ML files in the future. So this is the basic elements of SED-ML. It's these 4 things. There are models. You define a model as a reference to a model. So here we have an SBML model, a file that you claim what it is. Then you can define a simulation, without referring to a particular model. This simulation one is just a uniform time course from 0 to 5 with 51 points. And then a task connects those two. So a task would say, I want to run simulation 1 on model 1, and then you actually run a uniform time course using that particular model. So obviously you can have a stable of models, you can have a stable of simulations and run them all, however many combinations you want. Outputs, then if you want, you can have plots and reports. Plots are the figures. Reports are CSV or HDF5 files that just have data in them as a result of your simulation experiments. You can also, I should also explain, so this text that is on the screen is something called phrased ML, which our lab developed. Kiri and I sort of were like the main programmers on that several years back. That basically translates SED-ML into a human readable, human writable form. And it doesn't cover everything that SED-ML can do, but it covers the basics. So you can sort of create SED-ML relatively straightforwardly without having to worry about stuff. There's actually a part of SED-ML that I haven't even mentioned at all called data generators that are completely elided in phrased-ML, because they aren't really part of the semantics per se. If I have like S2 divided by the compartment, meaning that I want the amount, I want it in concentrations instead of amounts, then that's something that would technically, in the innards of SED-ML, need a data generator that says like produce this and give it this value and do that. And phrased-ML tries to like hide all that from you, so you don't have to worry about it. You can do more complicated tasks. You can do which are mostly repeats. You can loop over a series of things and run your experiment several times. You can run, in this case, task 1, setting S1 to a few different values. Here, I should, instead of pointing at my screen, I should use my mouse. You can use S1, set S1 to 1, run the experiment, set S1, then set S1 to 3, run the experiment, then set S1 to 10, run the experiment again. Or for this one is a uniformly distributed set of numbers from 1 to 10 with 50 points in between. Are there any questions so far? Because I don't want to go too fast. Anyone for whom this is new? Yeah.
What about parameter settings and like which variables are to be output?
So the outputs are this sort of thing. You say I want to plot time versus S1, S2 or something.
Oh, I see. You just list
So you just say that, and actually, and internally, yeah, internally, like if someone gets a SED-ML file and they want to know what to do, they sort of have to look at the results and then figure out, back calculate what they need to store. But in general, this is what you do. You have data generators that give you these values and then plots that produce the output that has them on it. And you can set things. You can set values. So just like this, repeat this task for S1 in this. You can set the value of S1. You can set parameters that way. You don't have to just set anything else. You can also set things in the original model itself. You can say like model 2 is model 1, but S1 is 5.
Cool.
If you want to do things like that.
Couple of quick questions,
Yeah.
Lucien. So on the next page, we have no, I'm sorry, it's the previous page then.
Okay.
Yeah, right. So when I see that line that says simulate uniform, now in this case, is uniform a model, or what is uniform?
Uniform is a keyword in phrased-ML that means a uniform time course. In SED-ML itself, it is a class. It is the uniform time course class.
Okay, and so what is actually happening there? Because there's no model referenced.
Right. It is just a definition. SED-ML is declarative in general. So this is declaring this is what simulation 1 is. When it is like a function definition, basically.
Okay.
So you're just setting up simulation 1 as a function definition that is simulate a model from 0 to 5 with 51 points, and that's all.
Oh, okay. All right, fine. I get what you're after there. All right,
and
fine.
then
Go ahead, you finish.
No, I was just going to say, and then when you actually like call it, it's sort of like this one. Run simulation 1 on model 1 is a task that is sort of like a line in the script that is simulation 1 parentheses model 1 or something. You're passing it model 1.
Gotcha.
Yeah.
Okay, so it's sort of, all right, that's interesting. Okay, and then the report time, S1, does that actually output like a CSV file or something else?
There are, if you have complicated input data, the data might be 3-dimensional. Like you might have one of these repeats. So you have like time course data for 3 different things.
Exactly.
And when you have multi-dimensional data like that, CSV isn't the best way of storing it. So you might end up with an HDF5 file instead. That's the way biosimulations has set things up. In fact, I think biosimulations always gives you HDF5 files if you ask for a report.
Okay, fine. So that's fine in terms of format. So the answer. Yeah. So what I'm hearing is that instead of a plot, we could just get the data.
Yes.
Simulation now. Okay, fine. Because that for some of this, that may be more convenient depending on exactly what we're looking at.
Right. And there may be times, in fact, where all you can do is get the report because the plot doesn't have enough semantics to sort of say exactly what it is.
Okay.
So you would need to, like, at least you can get the report, and then you can write something else that'll input that report and give a fancier figure using, I don't know, matplotlib directly or something.
Yeah, when creating these SED-ML files, I would recommend outputting everything that you can possibly output, because then the UI will allow the user to create their own custom visualizations using the output data.
Yeah.
So even though the
Okay.
paper might only, plot a certain species, if you include all the other species in the data output, a user can play around and create whatever visualization they want with those species.
Yeah.
Okay.
Yeah. And I believe
Fortunately,
that
it's like a
part will have
requirement.
already been done for you. Sorry, there's a.
Oh
Go
yeah,
ahead.
I was gonna ask, like, is that already done with the processed SED-ML files?
Yeah, the process SED-ML files do, like, all variables have like an auto-generated just list of like, here's all the variables in the model for the tasks that we have.
Yep.
But if you're creating a new task, then you would have to like do either run that script again or something else. And we can talk about how to do that as I feel like we should talk about those, like, as people come up with specific questions, like, I need to do this. Just know in general that there are a bunch of scripts that we've used to sort of do the basics for you here.
Okay.
Combine archives. So that's everything that I have to say, like, introducing SED-ML. Combine archives are zip files that are called .omex files. You just make a zip file and then rename it .omex, that have a manifest.xml file in it that lists its contents, and that manifest has a particular format. It will include the SED-ML file, any model files. It can have multiple SED-ML files. Any input data, it could include the expected output. It could actually contain the figures that you are trying to produce. Any auxiliary files.
The annotations.
Annotations, yes, it could contain annotations, and it
Please.
does. Yeah. So yeah, all the various things you might want can all be put in the Combine archive and listed in the manifest, and then can be manipulated by things like biosimulations. And different tools will recognize different parts of it, like Tellurium. I'll get to Tellurium later because I need to talk to you about that. Anyway, different tools might recognize some of the pieces, but not all of them, depending on what their purpose is. Speaking of Tellurium, so Tellurium is a Python-based simulation environment produced by the SAR Lab. It uses another thing produced by the SAR Lab called LibRoadrunner, as its core simulator, with a variety of wrappers and things to make that easy for Python users. It can read and write Combine archives. It also supports phrased ML. So if you want to, you can actually like write a phrased ML program and have it translate things to SED-ML for you. The only issue is that if you are writing Tellurium, if you're like actually running the experiment in Tellurium and you say like simulate this, then copy this data and simulate the other, that script is Python, and you can't convert a Python script to SED-ML. So that's the part that's going to be the most awkward part of all of this. Like, you might be able to produce the figure, and then you're gonna have to say like, all right, now I have to do exactly that same thing again in a way that that I can either write it in SED-ML directly or that I understand for SED-ML.
Can it do the reverse? Could it take SED-ML and produce Python?
Yes, it can.
Okay.
It's not the most readable Python in the world, but it is Python, and that's
Yeah, I mean, that might be helpful. Potentially, that's going to be helpful in terms of debugging to understand how commands are interpreted.
Yeah,
okay.
That's true. I didn't call out those functions specifically in the things I have today, but
okay, that's okay.
They're there. Yeah, I think it's called something crazy like convert Omex to Python or something. So
okay.
Coposi is another thing, is another simulator. It has a similar function to Roadrunner, but it does it in Tellurium, and it does it in a different way. It's a GUI. Essentially, if you want to model your SBML model with tables and menus, then that's a common way of doing it. Coposi is a pretty popular tool among SBML simulators. It does have a Python interface. I mean, it has a C++ interface also. It's not the easiest to use. Oops, I'm being recorded, but it's there, and it will presumably improve over time. The most interesting thing is that it can produce SED-ML. So if someone's already used Copasi to create a figure, then they can just hit export to SED-ML, and that will produce a SED-ML script for you. And we've been working with the Copasi developers so that it produces better SED-ML, because the initial SED-ML was producing didn't work all the time or often. And now it's doing much better. Next is Biomodels. So Biomodels.net was set up many years ago as an online repository. Initially, it's the SBML models. They are trying to sort of move away from only having SBML there, but it's like, I don't know, 99% SBML models at this point. Since it was set up so long ago, well before SED-ML was even conceived, it has the models, but it doesn't have what you do with them. So when a curator would sit down, that worked for Biomodels would sit down with a paper, they would recreate the model in Copasi usually, and they would recreate the figure until Copasi showed them the figure that was in the paper, and they're like, great, this is the same model. Then they would save to the SVML, and that would be what was the biomodel, was the SVML thing. But everything that they had done in Copasi to create the figure was often lost, unless they saved the Copasi file itself. And many of the biomodels actually still have the Copasi file there as an auxiliary file. So when they exist, we can still get the SED-ML out of it. And I just talked to Roman, who's running the Biomodels people thing right now, and they may have a bunch more Copasi files that aren't associated with particular biomodels yet, but were probably the ones that they were used. So I think I'm going to try to write some scripts to get the Copasi file, export the set of SVML, see if I can match it to one of the unmatched biomodels, and try to get that set up so we can sort of automatically do capture all the work that was done in the past with new SED-ML files there. So that will hopefully happen this week. He had mentioned them earlier, but it was sort of a low priority. But at this point, I'm like, I think that would be a good time to get those.
Sorry, what's the logic of that? What would you use the Copasi files for?
To create SED-ML, to actually create the figure.
I thought we already had the SED-ML.
No. Often we do not. I would say we have SED-ML in about half of the biomodels.
Hold on, though. All the biosimulation models have SED-ML files.
The biosimulation models do have SED-ML files, yes.
Yeah.
And and we have automatically generated SED-ML files for all the biomodels. But they all, if there wasn't one already there, then they just say simulate for 10 seconds, output all the data. And that's it.
Oh, you just want something a little bit more than that.
Right. So if you want to actually reproduce a figure that they didn't necessarily they didn't necessarily simulate for 10 seconds. They didn't necessarily even do a simulation at all. Maybe they did a steady state analysis. Maybe they did a parameter scan. Maybe they did who knows what. Okay. But they did, we know that they worked on it. We know that they produced at least one figure from the paper. So if we could find that, we would have a jump on all that.
Okay, I see.
We have been working on mostly sort of Jonathan Carr and Bilal and I, and maybe a few other people at the biosimulations have been working on sort of Biomodels curation project. We've been working on going through recurating all of the models and all the SED-ML files that they had. I think initially Jonathan went through and tried to run all the SED-ML files that were there, and 2 out of maybe 2%, or maybe literally 2 out of 1,000 or something, or 500 actually worked, and some of those worked accidentally because of internal reasons. Many of the problems were the same problem across many models. So we've been working both with Coposi to regenerate better SED-ML to begin with, and then also just writing scripts to add things to the SED-ML, so that now we have much better SED-ML, and in general, everything's been validated much more than it was. Biomodels has been focused on the models itself. The models themselves were generally pretty well validated, but all the auxiliary files that were there were not necessarily as well validated or anything else. Part of the goal of the project is to provide tools for curators going forward, and to provide links on the actual Biomodels website to biosimulations where they can run the simulations using those models. So the more models we curate to create actual figures, the better a site Biomodels would be for those when we do biomodels, because then they'll be able to actually, like, users will go to the website, they'll look up a model, here's a figure, click on rerun it in biosimulations, and hopefully it'll show them something good. You do your job well. Speaking of biosimulators is part of the center, organized by Jonathan Carr. Is it currently officially under different organism? Like, is someone else officially running it now, or is it not yet? Okay.
Still on the center. It'll need a
Wow.
separate grant if that would
Bilal
happen.
owns it.
Bilal owns it, yeah, pretty
Yeah.
much.
Well, he operates it, let's put it this way.
Yeah. The owner by default at this point. It's a place for curating experiments and simulators. I'm not going to talk a lot about it because Bilal's then now going to show you, like, literally what it is. But in general, its purpose is to allow users to run pre-existing experiments that other people have created, or to create their own using a stable of simulators and Docker images. So
But that's not biosimulators, that's biosimulations.
Bayesians, that's right. I should change
Yeah,
that.
the tools are the simulators, and
Right.
then the output are the simulations. We thought that
Yeah.
that was a pretty clear name distinction, but it's caused a lot of
It,
confusion.
it did cause, it's a legacy decision that ended up causing a lot of confusion.
Yeah, but we thought that was, like, the most self-descriptive name you could come up with. The
Right,
simulators are the tools, and then the simulations are the output. But
right.
that's caused
It's
confusion.
one of those things that, like, if you think about it, then it makes sense, but for some reason it's easy to not think about in the first place.
Yeah.
So, and biosimulators and biosimulations is organized around the Combine archive. So everything is sort of centered around there being this zip file with a manifest and a SED-ML file in it that will then run on the model, also in the thing. Everything is contained in the zip file. And all you need to do is run the SED-ML and then you're good. The only other thing I want to say is SED-ML itself is under development. We hope to make it more compatible with workflow languages. I told you why we don't want SED-ML to, why we want SED-ML instead of just a workflow language, but you still want to be able to translate SED-ML into a workflow, because you want to be able to say, like, this is the semantic, what I want to do. And then you want to be able to translate that to, this is how you actually, these are the commands you would actually run. Elements should have more clearly defined inputs and outputs. One of the things our lab is thinking about, at least, and I've done some forays into this, is to make sort of a simulator agnostic Python API that would hopefully allow the easier creation of SED-ML files. The idea would be, if, as you called all of your simulations, it's recording what you're doing in the background, and then you say, like, all right, export to SED-ML, and it says, like, all right, you've done this, I will give you a SED-ML file of the things you've done. That doesn't exist yet. If you have ideas as you're doing this, like, it would be really nice if this happened, let us know, because those are all design things that would be really useful to know what would be helpful. And if there are things in SED-ML that it can't do, let us know. Right now, the basic things are, it can simulate, it can simulate stochastically, it can simulate, it can simulate deterministically, and it can calculate a steady state. And in the very brand new version of SED-ML, it can now do a parameter scan, which it didn't. No, parameter estimation. It can do parameters. Basic things can do parameter scans with a repeated task. Parameter estimation is a whole new thing that you can do with input and output data. So if there's a paper that you're trying to curate that has parameter estimation in it, you should be able to encode that in SED-ML.
Yeah,
There won't be a
I
lot of
doubt it.
tools that do it. What?
I doubt it.
I mean, SED-ML could record it for you. Whether anyone can actually read it and
That's what I mean. Yeah.
rerun it, yeah.
Yeah.
Actually,
Exactly.
anyway, it's there. If
Yeah.
we need to do it, we can figure out how to do it. There's also a new generic sort of analysis class that should let you do anything as long as someone else knows what you mean when you say that. And I can explain that more if that comes up. So I think that is it. Are there any more questions about any of those?
You'll
things?
probably need an example. You'll probably have to go through a curation.
Yeah.
at some
Yeah,
point.
that's my plan.
Okay.
So
right.
so I just wanted to sort of set things
Yeah,
up, like
okay.
this is all everything, but also I'm going to show you the biosimulator website,
Okay.
and then I will actually go through a script that I wrote that
Okay.
actually does use
All right,
some
great.
of this to curate stuff.
The other thing that would be helpful, Lucian, is what would be the right reference to use for something that is, not a reference manual, it documents all the APIs, but something that gives you sort of a good, like introduction to SED-ML so that we could understand, from some documentation how to do it. Is there a how-to guide?
There's the spec. What more do you need more than the spec?
That's true. Everything's
Perfectly-
derived from the spec.
Everything's in the spec.
transparent.
You don't need anything more than that. Yeah.
Well, the problem is that the spec is also sufficiently vague that certain things are open to interpretation, and it's one of the things that has been made better in 1.4, including use cases at the end of the spec.
Yeah.
The best sort of
Best practices or
best
something like that.
practices, right?
Right.
But it's still one of the weaknesses of the standard.
Yeah. When Jonathan found SED-ML and started like asking questions about it, I think he filed like over 100 issues on the SED-ML issue tracker that were all like, this is unclear, or this is other part is unclear, or how do these two things work together? And everyone was like, and it was interesting because a lot of the developers of SED-ML just sort of assumed like everyone knows that, right? And it wasn't actually in the spec. So it turned out no one did know that. Or it turned out that like different implementations of SED-ML did different things, which also isn't helpful. So yeah, with L1v4, which is the latest version, level 1 version 4 of SED-ML is the latest specification. A lot of clarification was added, and hopefully enough. If not, then ask more questions, file more issues. We're happy to make things clearer.
Is there any
more
sort
than
of
half of the damn text is in red in L1v4?
Yeah, all of which is stuff I wrote, basically. So yeah,
Is
it's
there
like
any-
I'm probably like the world's expert on SED-ML right now because I literally wrote half the spec at this point.
Is there any tool that does sort of some kind of validation of
Yes.
SED-ML or
Yes.
okay?
Yeah,
Yes.
and that's again stuff that because biosimulations has been so focused on omex files and SED-ML, they've written validators themselves and then put them in Python packages so that other people
Okay.
can,
All right,
can
good.
load them and
Thanks.
validate them. So yeah.
So I sent a link. It's not documentation about SED-ML, but it is the documentation about our specific sort of best practices on top of SED-ML. So
Oh yeah, nice.
that's not exactly what you were asking for, but that is going to be something that can be helpful when making the figures and things like that.
Yeah.
So some of the things are just like using they're basically filling in some ambiguities with SED-ML. So things like using concrete XPaths have to be like the entire path and things like that. Yeah, that might be helpful to read through. Lucian, you said you actually created a script to do the curation. So I'm assuming a lot of like no one should have to actually manually write SED-ML.
One would hope, yeah. I have, so basically as part of, so as part of this, in temp biomodels, there's, I don't know, I think there's like 20, 25 different Python scripts that all do different things. And among those are scripts that run validation on these different issues.
Okay.
on the
Yeah,
different
so then
files.
most of that should be handled already.
Yeah.
Okay.
I mean, and it doesn't, to be clear, these are not things that go through and validate things. These are things that, like, in a programmatic way, call biosimulators utils,
Yeah.
and have those validations run.
Yep.
So they should be relatively straightforward. If you need something done, there might be some sort of Python script in there that does that already. It's not always obvious which one's which. And again, more than happy to answer any questions, or to have random Zoom calls with people being like, how do I do this? Is perfectly fine. All right, should we switch to Bilal for?
Sure. Yeah, so in terms of actually doing the validation, there isn't too much to know specifically about biosimulations. So let me just share screen here. And if I disappear, my Zoom sometimes crashes when I'm sharing screen. So here's just an example simulation run. Essentially, in order to do the curation, the two things that we'll really need to know are the ID of a run. So a simulation run.
Can you make the font a bit bigger?
Yes. So essentially,
There we go.
every simulation run on biosimulations is given a unique ID. As we are curating, we can then submit the omics archive to biosimulations and just record that ID as essentially the curated version of whichever project. So the output essentially, this is what it would look like for, for example, a model that has not been curated yet. So you can see that the user can then go and select, the various data that has been output. I'm just going to submit all of them so we have a figure to actually explore. And then, they can go ahead and then view this chart. So the main purpose of this, and Herbert, correct me if I'm wrong, is going to be to essentially create these charts so that when a user opens up this page, they have a list of figures as they correspond to the paper that they can select.
Yep.
So yeah, this is an example essentially of the way that we've set up the SED-ML right now. It outputs all the data, so that gives the user the ability to like actually select, the inputs and then graph them. The goal is to have this automatically done for all of the figures in a paper, so it can be reproducible. So things that are going to be helpful here is every simulation run should have a log. That's going to show you essentially the tasks that were run and if there were any errors. We're not doing metadata right now, I guess. That can be something we can focus on later.
Yeah.
Right now, we just want to get the figures working. Okay. Yeah, so the log here will show you, essentially everything that happened on the back end. And, one example right now, like we're not focusing on metadata right now, but I can at least show processing the metadata for this failed because there was an error in the file. So from here, if you now want to download this, you can go to the files. This is going to be a list of all the files that are present in the archive. So you can download any of those files individually or just download the entire project. Click on that. From there, you can open it up, make any edits, run it again, and see if the curation that you were trying to do worked. I can show
Sorry,
some.
what was that workflow? So this is the kind of thing they may do, right? They'll download the project.
Yep, and then that's just going to give you a Combine archive.
Yeah.
Any changes and things that are being made, you can make them and then just re-upload.
How do you re-upload?
So to actually go and upload this, you can go to run a simulation. From here, you can either provide a URL to an archive or actually select the Combine archive. Here's one. Scroll down, it should automatically detect the format and algorithms, and then from there it'll filter the tools that you can select to run these. I think Tellurium and Copazi would be the ones we want to run for biomodels. Anything else? Or yeah, so either pick Tellurium or Copazi.
So does it when you upload, does it then replace the old one? Does it?
No, so it's just going to assign it a brand new ID. I figured that was the simplest way to do handle curation, is that your run has an ID, and then that ID you can save to an Excel file if that's what we want to do.
Now, what happens though when somebody searches the paper and finds it? Do they find two then attached to it?
Yeah, so that we are going to handle later in terms of actually publishing these. So none of these are going to be published right now. As long as you have the ID, you'll be able to view them. And then once we have like a definitive list of biomodel 01 corresponds to this ID, et cetera, I'll go through and just make those ones public.
Okay.
Yep. Yeah, so you enter a name, submit, and then here goes. Now you're given this new ID, and that's the ID of this new thing that I've uploaded.
Yeah.
I can show some of the example ones where we're trying to, can show essentially the figures that we're trying to get to. I have no idea which one of these are what. I think this one has a nice little figure. So over here, like when I go to actually select a chart, this is what we're trying to get to. So you can see like here, this SED-ML already includes a pre-created figure that corresponds to figure 4A. So when I click on that and I hit view, it actually shows me the figure as it would appear in the paper. So that's kind of the overall goal. So
Yeah,
this
that's
would
the
be the
objective.
easiest way to test and see if the steps that you're doing worked. Just upload it and make sure that it actually shows up here.
Although,
Yeah, that's really all there is to the biosimulations part of things for curation. Does anyone have questions or?
I had a
So
so
what's the, I'm still confused as to what the success rate is in terms of how many models in from biomodels that are now available in biosimulators actually have biosimulations that correspond to figures. So here's
we
one.
don't know. I mean, like that's the first answer. The first answer to your question is we don't know, because that's exactly what we're trying to figure out.
Gotcha.
All we know is that some biomodels have Coposi files, so we
Gotcha.
hope that the ones with Coposi files have some chance of producing a figure. The ones
But they might
without, they might not. Yeah, they have a chance of producing a figure from
But
the paper.
the other question I think he's asking is how many models are there on biosimulation?
Right now, we haven't
Bucket,
uploaded.
bucket.
They aren't there, but we have OMX files that you can put there and like upload if you want to say.
So
I
they
would
won't
like
have
to
to
say-
down. So people won't have to download anything from biosimulations.
Yeah, that's all in a Git repository. So
Oh,
can
right.
just
So they'll
working on
be picking models from the Git repository, doing their thing to them, and then uploading.
yes.
Oh, okay.
Yeah.
So I can actually show. I'm sure, Lucian, you're going to go into this in more detail, but I can show what the Git repository currently looks like. Actually, I wanted to share my
It
screen.
might be worth producing a chart, a workflow chart, so people can follow.
Yep.
First
I
go
made
to this
one.
GitHub page, pick model, and of course we
Here.
have to make sure that people don't pick the same ones.
Yeah, exactly.
I'll have some way of dealing with that. Then the workflow chart says, create the figures, etc. Yeah.
So this is like I've have the repository kind of opened up in an editor here. Let me see. Can I zoom in on this? Yes. So in this repository, Lucian created a folder called OMX files, and this contains essentially all of the various OMX files. I'm not going to get into how to actually edit these, but let's assume for a second that every time you make edits, they're regenerated, they show up there. We can actually then go ahead and submit them. There's a Python script in here, so you can do just Python. The name of the script is run project on biosimulations, and then pick essentially the biomodels ID that you want to run. So let's say 9. It should then go ahead and submit it. It'll give you the URL, and you can then open that URL up.
Sorry, I didn't understand that at all. What are you running here?
So there's just a very simple like wrapper script that I've included now in the repository, which will take the already existing Combine archives and then submit them.
Submit them. We don't want them submitted though until they're curated, right?
It'll upload them to run biosimulations and output a link for you, so you can open it.
They won't be public. They'll just be there.
No. We need a very clear workflow here.
Right. So
yeah.
Herbert, there's two separate steps here. One
Yeah.
is you're running things on Run Biosimulation. The other thing, you pick a run that you publish to the projects page.
Yeah. So I figured
Yeah.
to
So
simplify
this is just about running, and doing it scripted and using the API to submit instead of going to the GUI and plunking in the omics.
That's what I
file
mean.
and
Yeah,
all
this
that.
is that's what I mean. Got to make the workflow really clear. Remember, most people on the call have never seen this stuff before. Okay, so it's really important that we have a chart or something. Go through these steps. But the moment it looks like something like download an omics file, then they have to use some tool to create all the figures, get them all working. Then they use this Python run thing. Is that right?
Yeah, so that's essentially the thing is as long as you can edit the omics file, you then upload the omics file to run by.
Okay, so this is the last thing they would do, right?
Yeah.
Okay.
Unless you wanted to, am I not coming through?
Unless, but
Yeah, no, unless you
maybe.
wanted to take, unless you wanted to just use biosimulations as a place to look at things. Like you can view things on your own. You can load them into Tellurium or something, but you may also want to just play around with the GUI and turn on or off things all over there as you're.
So I was envisioning like if you're trying to reproduce a figure, you can first run the project and then go and like actually in the chart select the things you want to see and see what you're trying to get to. It might be helpful to do that.
Hold on, you're suggesting they do all the SED-ML edits on biosimulation bracket S bracket.
Like again, I'm not actually very familiar with SED-ML myself, so
Yeah.
I was under the impression that the curators, at least from like the New Zealand team, for example, has their own sort of already existing ways of working with SED-ML. So we're not like I'm not going to be making any recommendations on how to do that. This is just like once you've made your changes, you save the omics file. You can very quickly from the repo just upload it and run it and see if it worked.
I mean, presumably they'll have checked it on their desktop first to make sure all the figures look like the figures.
Sure.
And then they upload it, and then they just do another check to make sure it looks the same on biosimulation.
Yeah. I mean,
bracket,
if you
S bracket.
Yeah, like for people who might not have a sort of setup where they can do this on their desktop, it's just another way to like the easiest way that I can think of for me because I don't really work with SED-ML is just upload the archive and it comes out.
A lot of people here might be lost, but I think it's important to create a workflow here for everybody to follow.
Yeah, I mean, that was sort of like the purpose of my email yesterday was because I originally had thought that it would be the curators from New Zealand who are all very familiar with SED-ML. So in terms of of the SED-ML, Lucian says that he's
I have that'll be next.
From my point of view, the workflow really seems like what Hubert said, right? We access one of these omics archives from that spreadsheet that was sent.
Yeah.
We download it on our local machine. We play around with that on the paper, try to get some stuff. When we get that, we export the SED-ML and combine archive, and then re-upload it to run biosimulations, and then that will give you an ID, and then you can put on the spreadsheet that this has been curated.
That sounds like it, yeah.
Right.
So that's all that
we can write that up.
Yeah.
Also,
how to actually edit the SED-ML is that's a whole nother.
Yeah. That's a whole issue.
That sounded good, and you should write it down quick.
All right. So that essentially covers the biosimulation side of things. Pretty simple. I guess one thing that might be helpful. Again, this really depends on what your personal method of editing SED-ML is, but if you do come here and design a chart, so I'm just gonna do like a basic time series of a couple of things. I'm not entirely sure.
We're still seeing your Python screen.
Yeah, we're still seeing your Python screen if that's what you want.
Just stop my sharing. Start. Yeah, so if you go on biosimulations and actually select a chart and create one yourself, there is this option here to export in archive, which I think code this into SED-ML. I'm not positive. So this might be something to try when you're curating if you want to see whether like it might provide some method of putting this into SED-ML if
Mhm.
you want to try that way. I'm not positive that it's actually encoding it into SED-ML. It might just be encoding it in Vega or something. So check
Okay.
up on that. But yeah, that's all I've got.
All right.
So for someone who's really not familiar with these tools and would like to be able to contribute to this, I think there's some significant gap between the information that I've gotten so far and being able to actually do it.
That's next.
Okay.
That's
All
part
right.
3.
Fine.
So,
All right.
all right. Is there any just basic understanding things like what is going on, questions over anything we've covered so far?
Oh, do I really have to do this?
Like,
Right.
what have I
Yes.
gotten myself into?
You do.
Yeah, that was the thing that's held us off from doing it for so long, is that this is just inherently a manual curation problem. Like this is not something that we can, really automate away.
So just to tell people the workload, I'm hoping everybody on the call, or most of us on the call, can do 1 or 2 a month. So give yourself one afternoon every month or something if you got the time, and then curate a couple, and then at the end of 10 months hopefully we'll have 100 or more.
So I think there's not really that much effort to curate many of them, except of the fact that it really is manual, as pointed out. But you start with already having an omics archive and a SED-ML that's been run there, that executes with the right parameters, and then depends on where it came from. Often you have like the default on display, just pick every variable to display. That's generated output. So you just look in the paper and say, oh, figure 1 is calcium across time plus IP3 across time. So you just take that plot definition and eliminate the other ones, and there you have it, and rename it as figure 1. But then you may discover that for certain things there is no task and simulation available, that on figure 4 they ran it with a different parameter and something else happened. So that will be a little bit more involved, but it's again just encoding a model change and another task in SED-ML. So I think it is not that complex, but it's tedious.
One issue, I guess, is a lot of these papers would have like bifurcation analysis type stuff, continuation plots.
Right, and if we don't have something for that, that's it.
Yeah, if you can't do it, you
Okay.
can't do it. Right? If you come across a figure and you think, oh, there's no way SED-ML can do this, then don't do it. But
So-
just
but
make a
no,
record of which ones you haven't done, I guess.
record
So
that you can't
here's
do it
the-
and why,
Yeah.
right? And
Okay,
because then
so-
someone else I might be able to come up. There is this generic analysis class in SED-ML that you could theoretically like make a bifurcation analysis doing, and then just put in what inputs do I need, and like, all right, do the thing, and then you have to write then the simulator that is going
The
to take
main
that
thing
as
that's
input.
missing here, Lucian,
Okay, yeah.
is the ability, and that's part of our framework that doesn't have this yet, the ability to process one Combine archive with multiple simulators. So right now, you have to pick a simulator, which in the contract is that the simulator will execute as many tasks as it can. Depends on what algorithm it supports and whatever, but nothing prevents you to have a Combine archive with 5 tasks that, there's a spatial one that only VCell can do, and there's a bifurcation diagram that only XPP can do, and the other ones run with Capacity. And you can do that right now with run biosimulation by actually submitting it to the 3 simulators, but you're going to get a disparate set of results, where you're going to get the results of the first 3 tasks from Capacity, and the 4th one from VCell, and the 5th one from XPP in a different simulation run. So hopefully it will be one of the things that we can do in the near future to say, use a combination of simulators to run multiple tasks from one SED-ML.
Yeah. All right, let me go through my little script thing. I will sort of show you what I would do and what actually I did with a particular file model. I was
You
hoping
might
I could
as
share my
well
whole
just keep
screen.
doing it instead of getting all these, training all these people to do it.
Right. All right. Here is, so this is the spreadsheet. So I went through, I've been looking at model 882 recently because I was using it in our workshop thing. So for my
Lucian,
model,
I sorry to
Yeah.
interrupt so quickly,
that's
but
okay.
I noticed that when I do a query in biomodels for SBML models that are ODE models with differential equations,
Okay.
there are differential equations. When I do that query, I get 463.
That's because they have not been properly annotated.
Oh, okay.
I
Okay,
think.
gotcha. Okay,
Yeah, I think
and
there's
this list that you have here is, how did you obtain this?
This is the list of all curated biomodels, like literally all.
Okay,
of
so you
them.
do all curated. So presumably some of those may not be ODE models.
Some of them are
models?
not. That's true.
Okay,
There
gotcha.
are a handful that are FBC. There's a couple
Okay.
that are qual. But yes.
Okay, all right. Just some clarification because it's a huge
Absolutely, yeah.
difference. You have like 2x the number of models that I find that are,
Right.
you know, ODE models and manually curated.
Yeah. Yeah.
So we've already made like quite a few changes to the model. So our GitHub repo is the source to get the original Omics files, not biomodels.
Right.
Can you and so could you put your repo in the chat?
Those are all links in these links here in the temp biomodels thing. These are links to the temp biomodels, but I can.
Yeah, no, I
the
actually want the
general
repo
thing
because
too.
I want to get all the models.
Right.
Okay.
Oh yeah, it's right here.
Oh, temp dash biomodels. Is that what I should be looking for?
Yep, that's it.
Okay, thanks.
There you go. Okay. So where am I? Oh yeah, so temp dash biomodels is the one. So this is the repo. It's probably simplest if you just check this out. The organization of the repo itself is that the original directory contains everything that is currently on biomodels.net. These are all the files there, or these are the files that you get when you ask it programmatically to download everything for you. The main model, there's a couple different versions of the SBML model, the URL and URN. One of the things that we have done as part of this temp biomodels thing is to say, like, let's just use the URL version and drop the URNs because URNs were a thing and they aren't anymore. So you don't have to worry about them.
Oh, that refers to the format that's used like within the file for references?
Yes, the annotations are done with URNs or the Miriam : this : this as opposed to a URL, which is HTTP something. So all the annotations could be annotated 2 different ways, and they were like, I don't know which ones people are going to want. We'll do both. And now we know which one people want because over time we've sort of settled on the other format. So that's the original. Manual fixes the directory is everything that we couldn't change programmatically. So a few things are different between this one and the original. If you create a SED-ML file by hand, we'll probably end up putting it in here so that that is then used as the basis for things going forward. And I might even create a workflow for people for that to see how, depending on how that works. But anyway, and then we have the final. Everything in the final directory is automatically generated by some GitHub actions on this thing that take the manual fixes directory, take all those 20 scripts, process everything through it automatically, and output these directories. So here we have biomodels 01. You'll notice that now we only have the _URL. We notice that there is a SED-ML file and there didn't used to be one. And then everything else is generally unchanged, except that those files have now been actually validated where they weren't before.
Do they have to worry
are-
about this kind of thing?
Say that again.
Do they have to worry about this?
They shouldn't have to worry about this. It's just like explaining like what is there, why is it here. So if you want, so it's the final directory. So there's two places. There's the final directory that has everything, and then also, I just the other day wrote a new script that takes those final directories and creates OMX files from them. So this biomodels 001.omx is a zipped version of final/biomodels 001.
This is what they actually need.
And this would be what you could
Download it.
use, except that, well, I mean, if
No
you want
ifs
to
and
edit the
buts.
SED-ML file, it's right there in the final directory, right?
Don't give them
Don't
options.
worry about it. It's like it's
Don't give them
already there.
options. Give them very specific. Just do this, nothing else.
Just do this.
You'd be totally
Anyway,
confused.
this is what you
otherwise.
need if you want to use biosimulations. If you want to use your desktop, you might want to use the final directory because everything's already out, right?
Yeah, again, this comes down to how are you going to edit the SED-ML, which is
Right.
Yeah. So
very
what do you
common.
recommend, Lucian?
I
And then
wrote a
just stick
script
with
that shows you how to do both, and then you can figure things out. And as people like, I don't know what people are going to be more comfortable with. I sort of know what I would be more comfortable with, but I
What are you more
sort of
comfortable
medium.
with?
I think I would be more comfortable just editing the files that are not OMX files.
Okay, let's
zipping
do
them
that
up and
then.
yeah.
Just give them
Anyway.
one. Just give them one option. Otherwise,
Okay.
people get
Otherwise,
lost.
it gets too confusing. That makes
yeah,
sense.
yeah,
Sure.
Yeah.
Use the final directory. Do
So-
stuff from there, and then
So this has got to go into the workflow, right? Go
Yeah.
to final directory, find XYZ.
Yeah.
Okay.
and then like put the SED-ML file somewhere that you create, and then we can do the rest.
What?
Um,
What was that last bit? That's good.
I think like as people create SED-ML files, I can sort of incorporate that into our scripts and create the OMX files out of them. We should be able to upload them to biosimulations.
Well,
easily also.
totally confused now. Anyway,
Okay, never mind. Well, I'll write up the specifics.
Yeah,
I'll
be
show
very
you- here,
specific.
let me
yeah.
go on and show you what I actually did. So where was I? Here, curation status. I picked Biomodel 882. I'm like, great, here we go. This is a link to the actual OMX file if you want to just grab.
So
the
you
OMX
should do
file.
exactly what you think they should do, but don't go off in any tangents, okay?
Okay, got it.
Right, yeah.
I decided to pick 82. I go
Right.
here to curate our name and I put my name in it. And that
Okay.
means that someone else shouldn't. And then I switch this to begun,
Okay.
stalled or finished. And then
Okay.
like after you're done, like you can put comments over here.
Okay. That sounds
So,
simple.
One
Now-
question on that
Yes.
field, Lucian, if I want to say, oh, I tried this, but I don't want to really do it, can I just erase it?
I would
Or,
assume
or
so.
do I
That would
have to,
probably
or
be
do
the
I need
simplest,
another
yeah.
option
I mean,
for the select?
feel free to put comments over here, like, hey, like, you know, like, whoops, this is an FBC.
Yeah, but what happens if somebody thought
Oh.
it was too hard for them and they wanted to try another one?
Right, that's fine. Then
What-
then
what
just
do they
put something
do?
in the comments, like, this is difficult.
Can't they
And then
just
delete your name from the
zero
curator
it?
name.
Can I,
just
can I,
zero it out?
Yeah.
can I erase begin?
Yeah,
or begun?
exactly.
Yeah, sure. You can set it
To
to
what?
anything.
No, begun.
Line 861.
You should be able to
column-
change that to just delete. Yeah.
Okay, great,
And then you can
thanks.
zero out your name as well.
Yeah.
Right, got it. Okay, thanks.
Yeah.
That
and
may
w-
be an important part of my workflow.
It's like backing out. It's like, never mind.
Yeah, can't do that one. No, that one
Yeah,
either.
right.
I mean, when you look at things, I think it would be useful if you put in comments. So if you wrote up like, hey, this has some sort of whatever model in it, and I don't want to deal with it, and then so just write that.
Yeah, you could write a
in
little
comment
comment,
file.
I guess.
All right.
And then
good.
someone else can, like, when the next person doesn't have to doesn't have to go through the same experience that you did already. Just
Okay.
so.
Got it.
One thing, so I don't think anyone currently has edit permissions on this.
Oh, really?
Yeah, I think it's anyone with those link and
Oh,
view.
just view. Okay, here, editor.
Oh, right.
Done. All right. Okay, now everyone can edit it.
Okay, so what do they do now then?
Right,
They've
yeah, I'm getting to
picked
that.
a model.
Right.
Now what do
882.
they
I
do?
go to biomodels.
What
Here.
happened then? That was quick.
Okay, here. You know what I'm going to do? I'm going to go to
I'm
biomodels.
totally
oops.
confused now. You went from the spreadsheet to biomodels.
I'm going to biomodels.net. Right.
Okay, so you've identified your thing. Now you go to biomodels. Okay.
Right. I'm going to click on, I don't know, here, manually curated.
We can also generate this link and just add it to the
We
spreadsheet.
could do that too, actually. Yeah, that would probably be simplest. What I usually do is I'll click on a random one. And then
Well-
it'll give me the basic URL form. Like, oh, there's a random one was 125. I
see.
want 882. So I'm going to edit this to 882.
Okay,
Bonk. And there I go.
right.
So this should give me the link to the publication is one of the main things here. And it'll also tell me like generally what this is. This is a model of a zombie outbreak. And I have clicked on this before, and I don't think it helps because it's like, oh, it's a book. Great. So when that doesn't work,
What
just
is your
highlight-
objective? What is your objective here, Lucian?
I want to get the paper.
You want to get the paper, okay?
Right. So I will copy that, go to scholar.google.com, paste in the title of the paper, and here it is, and it's a PDF. Click. There's my
Okay.
paper.
So they need to get hold of the paper,
You
right?
need to get a hold of the paper somehow.
Right.
That's how I got this one.
Okay,
You won't necessarily always get things. So
right.
you can scroll things down here. Look, oh, that's a nice picture of the model. But you won't always get this, but you know. Here's some stuff. Here's some figures. Oh, look, it's a great figures. 2 lines that don't change. Here's another figure. Oh, a line that goes down, a line that goes up. Great. Okay. Something like this is probably in the curated version. So knowing that, I now switch to Python. I will make this script available to people so that you can.
Well, you haven't got the model yet, so that's what you're going to do next, I guess.
Oh, well, the next, I mean, what I did.
The model is linked in the spreadsheet.
Yeah, it's linked in the spreadsheet.
Can you show us what happens when you click on it then?
Sure. Or here, I guess I have to stop sharing this again. There we go. Let's go to the spreadsheet. Oh, hey.
Okay, all right. so
Click
you
on
go
the
82.
model,
I see,
click
yeah.
on this, and it
Okay.
takes me to this, and blunk. There we are. There I am.
And now what do you do?
I mean, what I did was I checked out the temp biomodels.
Yeah, well, you can't teach them
Repository.
GitHub.
What?
Can't teach them GitHub.
I just want to check. If I don't want to, if you don't want to know GitHub, if you know how to do, if you know about how to check out anything with Git, check out the repository and Git. It's simple. All the files will be there. If you don't,
Actually, could you just download the zip file?
yes,
whole
that's what I was
repo.
about to say.
Ah, okay.
So
do
the
that
link
one,
is
yeah.
goes to the whole thing. You can go to the Omex files. This is another reason to use the Omex files. Find the one you want. Edit the URL or click on a random one and then 882. And there it is. And I can click download.
So you're
and
recommending they download the Omex file?
it's either that or download the directory. Either one is fine.
Okay, but what you do with it later depends, right? Okay.
Yeah.
All right. So you're going to
Okay,
get the
so
Omex
say
file.
I've downloaded the thing. Great. Let me just see if it's possible to download a whole directory because it might be.
You could download the entire repo, but
I mean, it's gonna zip it up anyway, so at that point you can just download the.
Yeah. Is there a way to just delete directory? No. I don't think I would have to like
yeah,
download the individual things. So
Yeah,
yeah.
that's why the recommendation was. I think to just download the Omex.
yeah.
Download the
that
Omex
probably makes
file.
sense.
Okay.
Okay. Done. Great. Now we
So
go
you
to
got
Python.
you got the Omex file somewhere now.
You have the Omex file somewhere. Maybe you have the final thing itself somewhere. All right. Dun, dun, dun. All right. This is my manipulate SED-ML script. Has a bunch of stuff at the top and a bunch of comments about what's going on here as we go. And I'm going to step through this, I think. Oops, except that I already. There. There. All right. So I'm going to show you. So as it runs, I'm going to show you what's happening. So first we import a bunch of stuff. So Tellurium is your simulator, or it contains the simulator. Biosimulators utils and Biomodels QC both you can get them with pip. and they have these convenience functions in them. Libsed-ML, you may want to end up using. OS and glob is just for other stuff, for file manipulation. So this is where I set up a few basic things. That my sed-dir is where I actually, that's the directory with all the files in it. So anytime I use that, it means that I've sort of it's a pointer.
But at the moment, it's just got the omics file in it, right?
So that's the no, the omics file is this. So if you want to use just the omics file, then that's that.
Okay, that was the route you took. So
yeah,
they don't have to worry about the first two then. It's the third one they
sure.
need to set up. Okay.
And then I want a temporary output directory that it's going to unzip everything to and dump stuff in. So this is what so Tellurium has a function execute sed-ml. If I have the directory with the sed-ml file in it, which you can do if you just unzip the omex file, you can. Okay, all you have, so to get the file, to get the directory, take the omex file, make a copy, rename .omex to .zip, and then unzip it, and then you have the directory. That's all you need to do to get sed-dir, basically. You can either do that by like checking out the whole repository, or you can just unzip it.
You need to write this down in the workflow as well.
Sure.
People will forget, for example, that omex is actually a zip file.
Yeah.
Right. So everything has to be written down explicitly.
Sure.
Okay.
You can do it programmatically, but it's much simpler than just like rename to zip, unzip it, done. So execute sed-ml. So there's our first time doing this. Oops, yeah. And here we get a figure. And look, we have a line going down, another line going up. We have susceptibles going down and zombie and removal coming up. And if we look at the paper, that's exactly what figure 3 was. And indeed, it's called figure 3 here. So that's a bonus. So like it has a name, figure 3, it has time, like the x-axis doesn't have a label, but okay. And we see the population going down and zombies and dead increasing. So that sort of seems like what would happen during the zombie outbreak. So you're probably good. So the basic thing is that you're like, okay, great. What I hoped for was the sed-ml file would have given me a figure from the paper, and it does indeed seem to basically be that. Just as another option, you could also just execute the Combine archive. If you have your omex file here, I'm going to do that, and it's going to do the same thing because it has all the same data. And indeed, here we go. It just gave me literally the same figure, and it gave me the same warning message also. You'll see. Basically, you'll see this warning message a lot because Copazi uses LSODA and Tellurium uses CVODE. So, and those are not related in how things are annotated, which I can get into more detail. And I actually asked if we could change that for Cassell in general. So that thing might be, we might be able to sort that at some point. And if we can sort it for everything all at once. So don't worry about this warning message. So now I'm going to do something else. I'm going to get a particular file out of the Combine archive. This is another, so if I know what the file name is, in this case, I know it's called moons2000.sedml, I can just grab it and then sedml will have the text for me. So bonk. And then if I print sedml, that's the actual file that is the sedml itself, in all its XML glory. That's just to sort of let you see what's going on. Similarly, I can use Biosimulator's utils to extract the files from a Combine archive. I create a reader and I'm going and run it. And this is where outdir comes in. Outdir was my temp 882 directory. And if I do that, then on my computer is now a 882 directory with all the files in it. So if I hadn't If I hadn't done that before, you can do that way. If you rename it to zip and unzip it, whatever, you can do that. Or you can do it this way programmatically, read the file, export it to something. That also works. So now this is just an example, like when you're done and you're zipping things up again, like imagine between line 37 and line 40, you've done a bunch of editing and now you're done. And now you want to you've changed the contents of the directory. You've edited this SED-ML file somehow, and now it's better. And now I want to make an omics file again. Then I run this little script, these 4 lines, which is create a writer, get a list of the file names, and then convert that to a list of locations, which is what the thing actually wants. And then this build combine archive function creates an archive out of that. Or it creates the description of an archive, and then the combine, the combine writer will actually create that. And after I run that, now I have a file. Oh, hey, I told it to stop here, apparently. So great. All right. Now I have a file 882.omex, which is a brand new file on my computer that is the zip thing from the directory. So, but like what happened between those 2 steps? There's a bunch of different things you can do to manipulate the SED-ML. Option 1 is you can use a text editor. You could open it in text. If you just want to do something simple, you can just change it. In this case, if we look at this figure and we look at the original figure, which I can't show you without like resharing my screen, so I'll just tell you, there was no line for the removal. Like there was no, like the removal is the dead, like the buried version of people in the thing. And all it had was people going down and zombies going up in the actual figure. So let's say we want to reproduce the figure exactly and take the removal line off. One thing that is the easiest way to do that, if that's all you need to do, is you open up the SED-ML file and you find, so here I've showed you this here, and it's the plot. You're looking for a plot 2D, and it has a list of curves in it. And curve ID 1 is susceptible, curve ID 2 is zombie, and curve ID 3 is the removal. I could edit the SED-ML and just delete that one line and it would be gone. And that's all I would need to do. Chances are you're going to need to do something a little more complicated, but maybe it'll be that easy. And in that case, this by far the simplest thing is just like literally just change the SED-ML file. Another thing you can do though is use libsed-ML. Libsed-ML is a low-level Python library for manipulating and creating SED-ML. It is designed, like all its objects and all its whole design is set up based on the SED-ML actual structure. So you will notice that one of the basic things that this has, so here we'll go through this XML itself. So we have the SED-ML object, and the child has a list of models with all the models. It has a list of the simulations with the one simulation in it. It is a uniform time course simulation with some annotations there. It has a list of tasks which combine the models, which is again, in this case, is exactly one model and exactly one simulation. It has all these data generators, which are complicated and annoying. And then after the data generators, it finally has a list of outputs, and that's where the plots are going to show up. So I can loop over all the outputs programmatically using libsed-ML, using something like this for O and range, sed get number of outputs, out equals sed dot get output 0, or O in this case, because O is the. So bonk, bonk, bonk. So if I look at out.
The thing is, Lucian, I don't think we can expect people to, can't expect people to have to write code like this.
I don't know. I'm just like, that's why I said, like, you might just want to edit the SED-ML directly.
Well,
anyway, so I'm going through a few different things. If
Yeah.
something here works, great. If it doesn't work, try one of the other things. I want to like.
No,
and-
but we could write a helper
And the reason
method,
why
you
I'm giving
know.
you several different options is because there's going to be all sorts of different things that we're going to need to do to match the figures.
We
Like
can't
sometimes it's going
expect
to be
people to
easy,
be
sometimes it's going to be hard.
libsed-ML experts, though. That's not going to
Yeah.
work.
Well, some people are, but anyway.
Who knows? Who knows? Put your hand up. Who knows libsed-ML?
Yeah, I know.
Yeah, see, that was what I thought was the original
The
audience.
majority.
Yeah.
Let me give you a better one. Who knows JLibSedML?
Oh,
Nobody. Okay.
no, okay. Because that's what. Well, don't you know that?
Well,
Yeah,
I
you
do
know.
because we're using
Oh
it.
yeah.
Yeah. See, there you
We're
go.
gonna have to create some helper methods like add curve, delete curve, add new graph.
I could see that. Sure.
You know, it's
Um,
not going to work otherwise. I don't think.
Well, anyway, here. So
Okay.
this is where I went through the like I did the very simple thing that you could do in text programmatically. I looped through things. I looked for the one named figure 3. I set a local variable. I'm going to loop through all the curves in figure 3, and when it was removable, like it is going to catch that. So it's like, oh hey, here what. Remove index is 2. It's the index 2. So great. And then I remove curve 2. And now I'm done with the loop, or actually no. There we go. So now I write that to a file, new sed.sedml. And if I execute that sedml, bonk, I get this. And indeed, I have one curve going down and one curve going up, and I don't have the dead on the list anymore. So that's one option. Here's another thing you can do. You can use phrased.ml, and I think phrased.ml might be the easiest for things that are out of left field or have nothing to do with the existing sedml. Phrased.ml is again, it's another Python thing. I don't have a version for Python 3.10 yet. I've been working on trying to automatically generate it for all versions of Python and to update it in general, because I have an updated version. I hadn't put it on pip yet, but the basics are still on pip and it still works with this very simple things. If you have no problems with it, I can fix it, and hopefully I can make it even better soon. So here, just like I was showing him the original PowerPoint, this is a phrased.ml model definition or simulation experiment definition model or description, whatever. We make a model using the model keyword called no zombies, because if we go back to the paper, the original figure was this figure, except that like people were a flat line and zombies were also a flat line because zombies started at 0. In this model, again, the model itself, you can see zombies start at 1, but if you start zombies at 0, they're going to be different. So I'm going to make a new thing. I'm going to say it is this model, this file name with zombie equals 0, and that'll change the value of zombie in the model. Then I define a uniform time course. No zombie sim is run sim on no zombies, and then plot figure 2 time versus acceptable and zombie. And that's it. I'm gonna have to import phrased.ml this time. The set working directory is important because phrased.ml has to be able to find this file. So you want to set the working directory to the directory where that exists. Then phrased.ml is just a converter. It converts this string to sedml. So bonk, and now if we look at sed2, over here. This is a new sedml file from scratch with a lot less stuff in it, but it has all the basic things. It has the new things. So here is the model. Here is the list of changes. It has the new value of 0. This is the target. This is the whole like weird XML thing about showing you how to get the zombie. and then the data generators, which are annoying, and then we have the outputs. Plot those two things, and it didn't even give them names. You can in the if you want.
I would say we definitely want to have names, just for displaying. It would.
Yeah, and obviously, like it came through anyway. Like the susceptible and zombie are actually in this figure, so we should probably do that.
Now you can't use this to remove the line from figure 3, though, I presume.
Yeah, you could do that also. You could, I mean, you'd have to create it from scratch, or you'd have to convert the SED-ML to phrased-ML and then edit that or something.
Yeah.
But maybe you could. Right now, converting the SED-ML that you get to phrased-ML isn't working because the SED-ML is level 1 version 4, and the version that's out there is only works for level 1 version 3. And if I manage to get a new version up, in fact, I have a version up and running for Windows. If you're on Windows and you want that, I have the thing you need at the wheel file. I just don't have it for Windows or Mac, or for Mac or Linux yet. I save it to a file, and then I can use the execute SED-ML thing again to do that. And look, this is my 0 zombies. And in contrast to the paper, the zombies start off at 0, but then it goes up anyway. And the reason for that is because in the model, dead always, like the population of dead always turn into zombies. There is just a rate at which all dead turn into zombies. So over time, some dead like show up. So I have failed to reproduce the figure from the paper. It mostly looked like the figure from the paper up through time 0.5. So then I'm like, geez, I don't know what to do. And I'm going to stop the share and go back to, oops.
Actually, I'm wondering, if you do something like that and somebody fails to reproduce a figure, there should be some indication in biosimulations to say, figure 3, and then note fail to reproduce.
Yeah, so here I can say like figure 2 might not be reproducible without reducing the time.
Yeah, but this should be
also,
indicated in
something else I noticed,
biosimulations.
the model claims it's the SIZRC model, and it is just not. It is the SZR model, and that's it. So there's a few things that are like, now that I've looked at it, I'm like, oh, there's some obvious things that need to be fixed here. And those are some of the things you may run into. And I think that's the end of my script.
Okay.
I'm pretty sure. So yeah, that was the end.
All right. So first of all, I'm going to ask, is there any questions? Probably everybody's terrified to ask them because they think I have no idea what he was talking about. But are there any questions?
Yes. Oh, we lost people too. So,
yeah, lost people. So I'm wondering how to move forward. So what I suggest is we rewatch this video and we come back in 2 or 3 weeks. And if people can have a go at something or try and see how far they get.
Yeah.
And then they can come up with questions and we can revisit this. Okay.
Yeah.
I mean, links to like GitHub repos for like phrased-ML and libsml and libsbml, sorry, and these things. I think that'll help.
Yeah, get a workflow drawn. Will be useful.
Yeah.
All right, everyone.
If you have Tellurium, Tellurium comes with it doesn't come with the biosimulators utils, but it comes with the other. It comes with libphrased-ML. It comes with libsed-ML. It comes with libsbml. All those things. So if it claims that those are dependencies, so you'll get them all.
I mean, I think there are a lot of potential pain points in this process, so we want to know where they are. I mean, I can see where some of them are, where they are, and in 3 weeks we'll arrange another meeting in 3 weeks. Okay. After you've had a go with 3 weeks. Well, I'm away for 2 weeks now, so 3 weeks time. It basically 2.5 weeks. The week after, the week in 2 weeks. Oh, hold on.
That's clear.
Yeah, I know exactly. Absolutely clear. So the week of 22 to 26.
No, I'm on vacation then, but
Oh, you're on vacation then.
Yeah.
Well, you're an expert anyway, though.
Oh, I'm also going to 26th actually. So
22nd.
let me see. Yeah, I'm gone the week of the 22nd.
Oh, okay. 29th then.
Yes, that'll be fine.
Are you away then, Anand?
Yeah.
29th.
Yeah, I'm away until the 1st of September.
Oh, okay.
That's that same week. But...
Okay, 29th, then set it up for the 29th then. And then everybody can have a go, watch the video. Don't be surprised if you get stuck. And then we want to know how you did and what are the issues and so on. And then one idea is then we can start writing helper methods to do the common stuff, right? Like one was remove a curve from a figure. Another one might be add a curve to a figure.
Yeah.
Another one would be create a completely new figure.
Yeah,
Kind of
I
thing.
think for the majority of them, we're probably gonna be looking at creating new SED-ML.
Things from scratch.
So some helper things just to create figures. If you think phrased ML is good enough for that, then just provide some examples of that.
Yeah, I don't know. It's
Can you augment with phrased ML, or does it just wipe everything out and do what it's
phrased ML just it converts from one to the other. So you could convert it to phrased ML and then edit the text.
but it can't augment, it can't add a new figure.
But it couldn't add a new figure.
Okay,
right?
in that
And
case,
we could try to write something that would combine two SED-ML files.
ooh. I would just add a little Python API, create figure with the following characteristics.
Yeah.
Keep it simple. Yeah. Okay then. Thanks. I think anything else? No, I think that'd be the best thing to do.
Well, Herbert, Bilal wanted to talk to you and me.
Oh yeah, okay.
Yeah, but that's unrelated, so you can do that after.
Okay. All right then. Cool.
All right, I will stop the recording. Thank you all for enduring
Thanks everybody.
this.
Yeah. We'll see you in
Cool.
3 weeks ish.
Hopefully it'll all work out.
Oh, 3 weeks. Yeah. 3 and a
Yeah.
bit weeks.
Cool. Yeah.